Welcome one and all. Thank you for joining us. What is this session all about? Well, we, myself, Fiona, I suppose you're part of it from St Andrews side of things, Noemi and Gibbon from Swansea. We're all part of a group called SWAMBA, the Scottish Welsh Applied Mathematical Biology Organisation, whatever we are. I can't remember the name. It's something very, very good, but I just like the term SWAMBA. And what I wanted to do was, quite often undergraduates aren't really given an understanding of how research works, what it's like, what do we do? You know, you only see your lecturers as lecturers and you never really get beyond that. So what this session is hopefully going to be all about will be, firstly, from my point of view, giving you a bit more of an idea of the structure of research. But then I'll be passing over to Noemi and Fiona, who will be giving you much more interesting viewpoints about what research they're doing. And that's all mathematical biology is. It's finding interesting research that you want to do. And you can do it anywhere. All universities do it. It's just that the University of St Andrews, Swansea and Cardiff are part of the SWAMBA group. So please don't think, you know, you have to go to one of these three universities. Oxford's also a fantastic university for this. We have Noemi went to a place in Florida, which she may tell you about, but there are places around the world that do wonderful mathematical biology. And really what I would say it comes down to is find somewhere that does something that interests you, because that's what's going to motivate you through your doctorate. Let me start off to sort of add on to that point about what can you do in mathematical biology and give you an idea that this slide is going to show you all the subjects that I found in a journal called the Bulletin of Mathematical Biology in the last 3 months. Okay, so as this scrolls, this in alphabetical order gives you all the things that were touched on in just 3 months. So you see from things from AIDS, breast cancer, dengue fever. That's of course part of the epidemics part of work. This one there, the effects of war on the Ebola outbreak. But hopefully you saw something in there that you maybe have thought, oh, that's interesting. I didn't know we could do that. And that's what this talk is all about, the idea of where you can fit. Because that's what I want to say to you, is that everyone can fit. It doesn't really matter if you're a pure mathematician or applied mathematician. There is somewhere in mathematical biology that you can fit. And I wouldn't even really say we think about it in terms of these ideas. It really comes down to a bit more about, my slide has gone. Oh, I've gone backwards, that's why. It's really all comes down to whether you want to be more general in what you say or more specific. Because I would say perhaps the more purer mathematicians take an equation or set of equations or system from biology and are much more interested in its structure. The equations may have been justified biologically. It may be a system of equations that look into breast cancer, or more likely, more recently, it was about the epidemics. And what they're looking for are the existence of solutions, the uniqueness of solutions, the bifurcation structures. What is in this setup, this system of equations that we can say generally about all types of equations, versus the more applied side, which I would say is more about specificity. If you're looking at breast cancer, maybe you're looking at a particular type of breast cancer, a particular line of breast cancer, and you're working with someone who has data on that particular line. So there you're testing a mechanisms. You're looking to predict what will happen in that particular line, and you try and generalize where you can. But you're not looking to say something about all breast cancers. You're looking for that particular type and understanding that particular mechanism. So that's what I would say we sort of fit on in terms of the spectrum. And one thing I should say now is these are my own biases, my own thoughts and feelings on mathematical biology. Fiona and Noemi, who are going to talk next, they will have their own thoughts and feelings. So hopefully they won't be too far apart from mine, but they may have different viewpoints. But what we just use as a case study as something perhaps very dear to our hearts is the idea of COVID. COVID fit onto the spectrum very well. All mathematical biologists had to work on COVID. It was law. If you weren't doing something, they would find you and ask you to do something. I tried to stay out of it, but they found me. The Welsh government found me and asked me to do something. In fact, Noemi, you were working, the group in Swansea were doing some fantastic work over there for Wales modelling. We did something very much more on the lower scale, and I wanted to sort of highlight that. And this is from my students. So my 3 doctoral students, Tim Osler on the left there, Josh in the middle, and Lucy, who's just finished her doctorate on the right. And they joined up to this NERC hackathon. NERC is the Natural Environment Research Council, and they had a load of data, a load of resources, and they set up a competition and said, we've got all this stuff about COVID, but we don't know what to do with it. Do something, and whoever does the best thing wins. And I'm very happy to say that the team here, they did win, but it was that very loose setup. It was, we have this stuff, we don't know what to do with it. We want to use it to help people with COVID. Do something, and if it's good, you win a prize. So they won £3,000 to share amongst themselves. And what they did was something you may say is actually very simple, very easy, but it was the right maths at the right time. And that's what I would say mathematical biology is all about. Mathematical biology is not a set of techniques. If you're an algebraist or a real analysis in real analysis or topology, there are certain theorems, certain lemmas, certain corollaries that you will all know and you will use day in, day out. Mathematical biology is being a jack of all trades. It's knowing what maths is out there and finding the right branch at the right time. So to give you an example of what they did, if you go to these links here, you can have a play around with this, it's still up. And what they did is they looked at public transport during the time when we were doing social distancing. And they were saying, okay, we have to stay 2 meters apart from all the other people. How many people can we fit on a train? And this is what they set up a nice little train structure, and they used a greedy algorithm to place people on this train, and they made a GUI for it, because then you can put shields in. These shields are plastic shields that stop the flow of air and allow you to put more people in. So that's what you've got here. You can change the length of shield, because then there was a question about how much plastic do you need to use? And then there was a question about, will we change the social distancing rules? So you could change how many people you could have on your bus, depending on how far apart they had to be. So that's what they did. Very simple. It's a greedy algorithm of fitting points, and because you could only sit where these points are. But so that seems very simple, and it was. But it won £3,000, and also it does highlight something which we hadn't realised initially. We use public transport for one reason, for being good for the environment. If you put more people on a train, it's better than individual people driving cars. But not true in this case. At the time of social distancing, you could not fit enough people on a diesel train to make it more environmentally friendly than a small family car. And that's what this graph was showing here. So even the simplest of mathematics applied in the right way can show you new ideas that you weren't expecting. And that, again, is the nugget, the center, the juicy core of mathematical biology that I love. Sort of to zoom out again of how mathematical biology is done. Again, I just want to give you how I see it working. It works as a cycle. You work with collaborators who work with you, and you work with them. They give you ideas, you give them data predictions, and it works back and forth. So you can jump onto this circle wherever you like, but let's jump in on the top left. You try and find experimentalists who have some interesting data, some interesting insights on a problem you would like to work on. If possible, get hands-on experience, because that way you'll know the limitations of your data. And that's really a crucial thing that many mathematicians, quite often myself, I don't necessarily have. I trust the biologists, the medics that I work with to give me the best data they can, and they do. There are limitations in there, but they will know, which I will not. So if you can, get hands-on experience. If not, work with the experts. At that point, you take their data, you take their insights, and you create a simulation, an in silico model of what they've done. And what you try and do is what I try and do is simplify it. What's the simplest set of equations I can get away with? That's called the bottom-up approach. There's a top-down approach where you try and model everything and you throw out bits, but hopefully in the middle you meet that you've got a set of equations that can reproduce reality, and that's your testing phase. Because unless you can reproduce reality, unless you can reproduce the data that they've got, you're not doing anything useful. But this is why mathematical biology perhaps tends to get a bad name, because once you've got to this point and you can reproduce reality, well, what have you done? We've already got the data. We already know reality. What have you done? But that's not the last step. The last step is once you have something that hopefully reproduces reality well enough, you can then use it to predict experiments that haven't been done. And that's closing the loop. Because once you can extrapolate results from the model, you can then say ideas that the experimentalists don't have. Hopefully then they use that to build their next experiments. And then again, the whole process starts again. They'll do the new experiments, you do your new model, you refine your model, you test it, and you predict, and you get some beautiful science coming out, hopefully. So in the last few minutes, I just want to take time to talk about something that I feel very passionately about, and that's the mathematics of pattern formation. I did this for my thesis, and I absolutely love it. And when I say pattern formation, it can feel a bit trivial, you know, working on zebra stripes or zebrafish stripes as we have here, or, you know, cheetah coat patterns. You know, who cares? It's not solving cancer. But really, it's the mathematics of how complexity is generated. So when we say patterns, we're talking about how your fingers form, what tells your fingers to form here, 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 and here. What creates the hair follicles at the positions they are? What creates your arms and legs? So although we look at pattern formation in terms of beauty, of pigmentation patterns on animals, we're really talking about any structure, the formation of any structure. What generates that? So there was a group in Japan, and they extracted some of these yellow cells, they're called xanthophores, and some of these black cells, they're called melanophores, from the zebrafish tail, and they put them on a dish, and they saw what happened. And they were amazed by what happened. I'm still amazed by what happened. These yellow xanthophores chase the black melanophores. As you might expect, the black cells, the melanophores, they create the black stripes on the zebrafish. The yellow xanthophores, they create the inter-white yellowy stripes of the zebrafish. So I loved this. I chose this to work on, and I extracted out these rules. I took reality and I abstracted it to give you the math. So I gave this Pac-Man model where we have these U cells, these xanthophores, chasing my little black ghost there, and they chase them, and most importantly, at an angle. That's what we see in the data. They don't chase in a straight line. They're not chasing off, going off in a direction. They chase in a spiral. So that's never been done before to my knowledge. So I extracted out these features, created the mathematical model of it, and here it is. You don't worry about it. There's just the equations that I extracted. But once you have the equations, you can then say, well, what can they do? And I can get stripes and spots, fine. That's not the interesting thing, because as I say, zebrafish, we know, have stripes. Here's the cool thing. Once you've got reality, you can then extend, see what your equations can do. And these are the kind of patterns that were available in these equations. These are live, active patterns. Now, of course, these are not biologically relevant, but here's the two ends that I was talking about. Going from the biologically motivated equations, the running and chasing mechanisms of these cells, which can produce stripes, and then playing around with them mathematically to understand, well, what can these equations do? What could biology do? So I'm going to finish there and give over to Fiona in a second, but I just want to highlight hopefully what I've done. Hopefully I've shown you mathematical biology is very cool. I love it. This is my everyday career, and I really have chosen a path which I love. I work on things that I play around with the mathematics, I play around with biology. There is the serious end of what I'm trying to do. I'm trying to constrain and understand the mechanism that biology used. But really, I'm just having fun with mathematical biology. I say don't worry, because I remember seeing some research as an undergraduate and feeling very intimidated. And what I want to say is, I've shown you an equation here. I'm not expecting you to understand it. What I would say is, if you've seen some things here that you don't know how to do, don't worry. I didn't know how to do them either initially. But that's the beauty of research. It's doing things you don't know how to do. And there'll always be something you can do, something useful you can say. So don't worry if you can't do it now. Hopefully you can do it in a year, in 5 years, in 10 years. That's the career of a researcher. There's always something you can do, some new insight you can give. And I hope I've motivated you. Hopefully you've seen something in my list of subjects or in the biology that I've shown you, something to motivate you. And even if I haven't, hopefully there'll be something via Fiona or Noemi, something you can see there, you go, that is really cool. I wonder why. Because I wonder why is where research starts. And with that, I will open up to any questions while Fiona sets her slides up. Are there any questions, or shall we go straight on to Fiona? No, if not, Fiona, please take it away.
Okay, I hope you can see my screen.
Yes, we can see your screen just fine, yeah.
Yes, so I want to talk a little bit about work on chemotaxis, which is a form of cell movement. And actually it's going to be based on patterns, kind of like the ones that Thomas showed there. So we're going to be looking a little bit more in detail of those patterns. And just a fun fact, a lot of this research was actually done in collaboration with other people that I work with in Northern Italy. So I get to spend 3 months of this year in Northern Italy. So that's another fun fact about research, is you get to go nice places and collaborate with people all over the world. So I'm going to be focusing on patterns in bacterial populations. So this is a bacterial biofilm forming. And we can see that these little bacterial cells, the darker areas, are a higher density of those cells. And we can see this very complex pattern start to form, and we see these kind of areas of patterns. So it's maybe a little bit more dense in the middle, and we have these kind of finger-like patterns coming out. And these bacterial biofilms are made up of a population of cells that are interacting and spreading out over this area. But these patterns aren't always of that form. We have very complex forms of patterns. We might have these kind of concentric circles, or these little dot patterns, or even more complex dots and circles, or these kind of shaded areas almost looking, where we have these areas of high density and areas of low density. And even things like spiral patterns start to form between these bacterial biofilms as well. So there's a wide range of complexity within these different spatial patterns, depending on the settings of this population. So one thing that's been shown with these bacterial biofilms is that chemotaxis plays a role in the formation of these patterns. So chemotaxis is a form of cell movement where cells are moving towards some sort of chemical or away from some sort of chemical. In the context today, we're going to be looking at where the cells are moving towards some chemical signal. And we're going to be looking at the case where cells are producing that signal to tell other cells that they want to come into that area as well. So we're going to look at a very simple mathematical model of this to start with. So this is based on a model that was made in the 1970s. And what we're going to include in this model is we're going to have some cell population, where at the moment we're going to assume all of our cells are exactly the same. And the cells are going to undergo two forms of cell movement. So they're going to undergo a sort of random cell movement. So actually from the video that Thomas showed, you could see that the cells that weren't quite chasing other cells were still wiggling a little bit. So that's why we're going to include this random cell movement, that they're kind of wiggling about a little bit. But then we're going to have this bias motion, or this kind of directed motion of chemotaxis, where the cells are going to be responding to this chemical and they're going to want to move towards it. So that's why we call this a chemoattractant, because it's some chemical that's attracting the cells towards it. So as I said, we're going to focus on a case where the cells are producing this chemical. So that's going to happen at some rate alpha. And then this chemical is going to decay over time. It's not something constant, and it's going to be used up by cells. And we have also that this chemical can spread out in our spatial domain. And the way that we can model this is we can use a system of PDEs, or partial differential equations, which you may or may not be familiar with, but don't worry too much if you're not familiar with these forms of equations. And they're actually very similar again to the ones that Thomas showed, where we have a term that relates to our random movement for our cells, which we're going to denote by n. And then we have this kind of chemotaxis movement. So we have something that depends on the cell density, but also on the gradient of this chemical that the cells are reacting to. Then our chemical equation is a second equation where we have this kind of random motion, which is given by what's called a diffusion equation. We also have production by the cells, and then this decay, depending on how much is there. And these models were kind of introduced by two mathematicians, Keller and Siegel, in the 1970s. So this work is something that's been going on for many, many years, and many people have taken these models and adapted them to make them more realistic and look at different applications of these kinds of models. So we're going to focus on a simplified version of this model. So we're going to just say that all of the parameters for the chemical are just going to be one. So we're simplifying this second equation and just really focusing on these two parameters, this kind of random motion parameter and this chemotactic sensitivity parameter, which is dn and chi. So with this system, we can do something called linear stability analysis, where we can use the kind of mathematical properties of these equations to really understand the system and kind of get an idea of what's happening from a more theoretical perspective. So if we do linear stability analysis techniques, we can look at what's known as the steady state of the system. So if we assume, first of all, that there's no motion, so we take out all of the terms that have this derivative with respect to x, and just focus on what happens at the kind of local level, then all that's going to happen is there's going to be no change in the cell population. This one becomes 0. And this one, the only change is going to be related to this n minus c. So if n and c are the same, then we wouldn't expect the populations to change over time. So within that linear stability analysis, what we can do is we can then look at what would happen to that steady state of the system where n is equal to c if we add in those spatial terms again. So if we take into account this diffusion and movement of the cells. And I'm not going to go into detail of it, but if you are interested, there's lots of work out there on this. What this tells us is that if this parameter chi, which is this one here, is larger than this dn, we would expect this steady state to become unstable. And that's when we get patterns arising. So from these mathematical equations, we can really get an understanding of this system before even doing any sort of simulations of this equation. So we're going to look at some simulations of these equations, and we're going to focus on this case where we expect patterns to arise. So we're going to look at some 1D simulations first of all. So we're going to set an initial condition and say we have some sort of random cell density. So this blue line represents our cell density over some space given by x. And the black dashed line is going to be our chemical concentration. So we're going to say there's no chemical at the start, but then cells will start producing it from those equations. So we're going to run that system where we let our two parameters be equal. And in that case, we saw very quickly that both lines became the same, and we don't really get anything interesting. But this was something that the maths told us from those equations, that if they're the same, we don't really expect patterns to arise. So we can then look at what happens when we change this parameter and make this a little bit larger. So if we make this a little bit larger, we see this time the lines go to the same, but then they start to change a little bit, and we get that this cell population, this blue line, starts to form this very large peak here. It's pretty much no cells here, and a little bit of cells coming in here. So we got something very different just by changing this parameter a little bit. And if we change this even more, we can get more peaks forming, and they start to sometimes join, and we get this kind of patterning arising. So this is in a 1D case, which is obviously not very realistic, but we can then scale these ideas up to 2 spatial dimensions, which is what we're going to do now. So we're now looking at the case where we have an X and a Y direction that the cells can interact in and the chemical can spread in. And our high density is going to be given by yellow and our low density by blue. And we're going to assume at the start we have a relatively uniform density of cells across our spatial domain. And then we're going to look at what happens if we let those interactions take place. So again, this is a case where our two parameters are equal. And from the maths, that told us that we didn't expect anything to happen. And we can confirm that with the simulations. When we let this run, nothing really changed in our population. Again, though, we can then increase things and see what happens there. So in this case, we get these little spots of high cell density, and then everywhere else is low cell density. So we have just these little pinpoints where we expect a lot of cells to end up. And again, if we increase this parameter even more, we can have more spots forming of these high peaks of cell density. So one thing we can notice about this is, in reality, when we look at things like bacterial biofilms, we wouldn't expect to see just very, very sharp peaks of cell density and nothing else anywhere else. So we have a little bit of a problem that these are not very realistic results. Another problem with these types of models is that actually, in some cases, we can get what's called blow up of solutions. So that's when we have numerical instabilities which means that sometimes when I run that system, the computer will tell me that you have more cells than what you started with, even though we've not included any sort of birth or death processes for the cells. So we're getting something that's impossible just by the way that these models are set up. So we want to think, well, can we do better? Can we make something a bit more realistic? And I'm going to focus on one particular case, but lots of people have done research on this and used different ways to try and get these types of models to be more realistic. So we are going to make a kind of simple change, and we're going to say, okay, instead of having a population where all the cells are identical, we're going to split our population into two. So we're going to have half of our cells at the start are going to be secreting the chemical. So they're the ones that produce the chemical, this N0 population now. And then the other population are the ones that respond to the chemical. So they're going to have this chemotactic movement. So we've got two populations now, N0 and N1, where the first population don't move via chemotaxis, but produce the chemical, and the second population move via chemotaxis, but don't produce the chemical. And we're also going to allow this switching between the two populations. So we're going to say that cells in population N0 can switch out into population N1, and the same thing can happen that cells in population N1 can switch out and move into population N0. And the form of these functions is something we're going to look at, and we're going to find that by choosing the form of this function, we can actually change the form of the patterns that we get in this model. So this is just a kind of initial case where we have different cases for the switching function. So case A is when it's constant switching. So it's just some value we choose. Case B is when we depend on the density. So however many cells there is tells you whether you're more likely to switch to one population than the other. And then a case C is when we look at chemically dependent switching. So depending on the local concentration of the chemical tells you how likely you are to switch between the populations. So don't worry too much about the detail behind these. All you need to know is that these cases are slightly different in different ways. But we can see in this 1D case, so the constant switching, we again get these peaks like we did with the previous model, but the kind of shape of these peaks is slightly different for these different cases. So sometimes we can have slightly smaller peaks, slightly squashed peaks, and we no longer get these very, very sharp peaks that we got in the previous case. And next to what we're going to do is we're then going to look at what if we have more extreme switching functions. So we have a little bit more variability in the population, that there's going to be a little bit more switching. And when we do that, these are 1D simulations again, we can get a wide range of patterns depending on the case that we're looking at. So this case here, we actually get something that doesn't really stabilize, and we just get the populations are fluctuating all the time, which is something quite mathematically interesting, but maybe not what we would see in reality, except in some specific cases. In this case here, we get very fat kind of spots of cell density. Here we got the peaks kind of similar. And then in this last case here, we actually got a case where the N0 population died out, and we were just left with the N1 population. So these were 1D simulations, and we can do the same thing and scale things up to 2D. So I'm going to show these three plots here, but in 2D. Hopefully the videos will play for that. So we see in this one, we have this kind of chaotic form where we just, let me play that again, where we just have fluctuations of the cell densities. Here we get these big spots of cell density forming, and here we get kind of target-like spots of cell density forming. But what's interesting is we investigated these different phenotypic switching functions a little bit more detail. And on the next slide, I'm going to show, I think, 9 different patterns that we got just by choosing different forms of that function or different parameters within that function. Now let me play that again. I need to slow some of these down, I think. So we see that we have these kind of sharp spots that we got in the previous case, so we can get those types of patterns. We got these more kind of labyrinth-like patterns of cell density. And what's interesting is we got these little ring-like structures. And if we think back to the very first slides I showed, these were kind of similar to some of the biofilm structures that we saw. So again, we're still quite far from biological realism, But by making that simple change in the model, we've been able to get a wider range of patterns, which helps us maybe understand where those patterns are coming from in reality. So I've talked a little bit about the bacterial populations here, but chemotaxis can drive patterns in lots of different populations, and cancer cells and things like that, they can use chemotaxis. Immune cells telling how the immune cells signal to each other to tell them where to go in the body. Chemotaxis is important in those processes. And things like populations of animals, we can use these similar types of models for looking at how animals, for example, are going to move towards areas with a higher resource. So I hope we've learned that math models here can help us establish when patterns are likely to arise in these systems, and that helps us understand those populations and how they interact with each other. And we can modify existing models. So as I said, these initial models were built in the 1970s, but still people today are using them and making them more accurate. And as people make them more accurate, they become more useful and more realistic to help us solve those biological problems. And with that, that's me. And if you have any questions, I'm happy to take them. Thanks for listening.
Thank you very much. Absolutely beautiful, Fiona. Really, I mean, one thing I would again also highlight about mathematical biology is the same idea can be used multiple different places. I mean, what you started off with was in a bacterial colony. I've been having problems like that in a yeast colony. So I need to get hold of your slides and have a follow some of those citations. I've not seen those before. Absolutely beautiful. Thank you so much. Any questions from the audience? Have to take them. If not, Noemi, please take it away. Oh, yeah, I should say, if you're not happy to put your microphone on, please do write in the chat. We'll do the questions at the end. Like I say, otherwise, Noemi?
Yeah, it's telling me that there's a failing share screen.
Oh, no, not a failing. Oh. Okay, here we go. Wonderful.
That's it. Okay, good. You can see the slide only.
Yep, yeah, it's good.
Okay, good. Right, so because I am originally from the city that Fiona visited, I was not happy with that. So I went to Florida instead. This is not to say that that city is not beautiful. Right, so I'll talk about work that I've done in collaboration with those people, and it's mostly centered around the problem of drug resistance in cancer. So if you look at this picture here, can you see my pointer? Hopefully, yeah. So the blue cells here are the nuclei of melanoma cells. They're quite small, quite round. And the bigger and more different looking odd-shaped ones are fibroblasts, the ones in green. You can also then see here and there some sprinkle of red, and that is a substance called fibronectin, as the one that is secreted by the interaction between melanoma cells and fibroblasts when the given drug is being applied. So specifically, these will drive resistance, will allow fibroblasts to help melanoma cells to escape the effect of the therapy. So this is the kind of problem that I'm interested in. From the clinical point of view, if we look at a patient in this case, it's for non-small cell lung cancer, so a specific type of cancer in the spectrum of specificity that Thomas mentioned, all the way to the far, I guess, end of the specificity. There's something we can do clinically, which is to monitor basically how big is the tumor in this specific patient. And this is a very typical response for giving a specific drug to the patient. So if we monitor over time, what is this tumor mass looking like? We'll start from some initial volume at detection, and then we apply the drug. And we see that typically we see a good response initially. So if you can follow this line, you see there's a good drop in the mass. Eventually, though, we see a rebound. And so it gets to a point where the therapy is deemed to have failed. So we typically will remove the patient from this therapy, a progressive disease. So here's the point where we will stop the inhibitor or the drug. And then typically we observe a regrowth of the tumor, but not only that, we actually see that the growth is quite explosive in its behavior. So it does actually make things worse. It looks like the therapy has made things worse. So we typically observe something called disease flare. Now, we sort of have an understanding of why this is happening. And to get a bit deeper into why we think this is happening, we actually need to consider two subclasses within this big tumor mass. And particularly if we consider the breakdown between the sensitive and the resistant portion of the tumor, then we can see that actually the cause for the response to the drug is actually onto the sensitive cells. By definition, those are the ones that can be affected by the drug. But there's another proportion, which is the resistant one, that is basically steadily growing, and it couldn't care less about the presence of the drug in any way. And at the same time, we can also see that disease flare is mainly driven by the sensitive portion of the tumor. Now, if we were to reintroduce the drug at some point later, what we would see is actually, again, because of the sensitive proportion, we would get some response again. But the long-term trend will still be a growing tumor. Now, to understand a bit better, so this tells us that basically this just breaking down between sensitive and resistant doesn't give us much information. To understand a bit better what's happening, we need to actually consider the fact that these tumor cells are not just a uniform group of individual cells that grow without limits and completely ignoring their surroundings. There is an intricate relationship between cancer cells and the environment in which they are growing. I'm going to, for the purpose of this talk, just basically talking about the environment, everything that is non-cancer as stroma. Part of the stroma is the fibroblasts, the green cells that you saw in the earlier slide, but there's different, all things non-cancer that will in a way have some sort of crosstalk initiated with the cancer. And this crosstalk will drive the emergence of resistance. So basically what happens in the course of treatment is that the tumor cops the normal tissue and is coopted by the normal tissue is coopted by the tumor, and it is activated by the treatment and is actually able to rescue the cancer from the effects of the drug. And so this means that we need to understand and then somehow be able to control this interaction, because we want to avoid the worst case scenario, which is that a tumor is already growing because it's resistant to a drug, grows even more because of the help it's receiving from the surrounding environment. So there's a lot of complexity here, but one of the things we can do is go to the lab and talk to the experts about actual cell biology and cancer biology. And a few things that can be done in the lab are something called in vitro and in vivo experiments. So I'll just briefly describe these and how they interface with the maths, but the basic idea is that we take some cell lines, and these are actual cancer cells that originally came from some patient, and we put them in a dish, in a petri dish. And we give them all conditions for them to be happy and go and divide. So ideal condition means we give them unlimited space and unlimited nutrient. And then basically we observe them in this dish, and we see how these initial cells that we seeded in the dish grow over time. Then we can do the same thing in an in vivo experiment. And what that means, we take the same cells and we can actually inject them into a specific location into the mouse. And again, observe over time how this initial mass is growing, constrained this time by the environment. So now there's no maximum capacity for growth because there will be some spatial constraint into where the cancer can grow. For example, we're seeding that in between some organs, so they will soon be confronted with constraints about pressure and where they can squeeze in, but also limitation in terms of nutrition. So for example, depending how far they are from the closest source of nutrition, which typically is a blood vessel, they might or might not be able to keep proliferating and survive and grow. So these two scenarios are actually quite crucial, and they're very possible to replicate from the mathematical point of view. So I'm going to start with the idea of the of building up complexity Thomas mentioned. I'm going to start with a very simple model, which basically considers a minimum set of dynamics and key players that we need to consider to actually replicate some of those dynamics. So this is called compartmental model. So what it means is that we keep track of different cell types, and we keep track of how the number of each one of these 4 cell types varies over time. So the top 2 compartments, they are the ones that are relative to the tumor. So we have cancer cells can either be sensitive or resistant. They have an underlying growth capacity. So they have a so-called growth rate. So each one of them, you see, they have their own, I'm calling these rho S and rho R, these are their own growth rate. Now, the drug that we're applying has as an effect, can impede the growth into the sensitive portion of the tumor, but will not affect directly the resistant portion of the tumor. Now, as a secondary effect, it will act on the non-cancerous components. So these are the other 2 populations that I'm considering. F stands for fibroblasts or normal non-cancerous cells. A stands for cancer-associated fibroblasts, or in other words, their reactive version. This is the version of fibroblasts that is able to talk to the tumor and help it into rescuing the effect from the effect of the drug. So as I said, this secondary undesired effect is that the drug will enhance the transition from normal to reactive. And in terms, these reactive cancers will give a boost to the growth into the resistant population. So basically, we can see that whenever we are applying the drug, we are limiting the growth of sensitive cancer, but we are indirectly and undesirably growing the resistant proportion. So it kind of looks like a no-win situation. But the good news is that all these things are transient. All these effects of the drug are transient. So if we were to remove the drug, which is now, I would like you to focus on the gray arrows, you can see that all of these effects are only transient and they are reversible. So upon removal of the drug, we can see some reverting back to sensitivity, and also the reactive stroma is going back to normal stroma. So basically, this is the perfect problem that we can throw at mathematics, because this is just about looking at what is the right balance in terms of how much treatment do we give to this cancer to kill enough of it, but to limit the undesired secondary effect, which is to enhance all of these interaction with the non-cancerous population that eventually will grow the resistant population. So I will not go through all of the details of the equations, but basically, now with some communication with experimentalists and understanding exactly what these experiments are representing, we can create a set of equations where we can pick and choose the sort of experimental setup that would allow us to create a version of these equations and quantify certain parameters. And we do this systematically one by one. And in the end, we have a model which is fully parameterized, which means it's going to be calibrated and has realistic dynamics in terms of the cell line that are specific to these experiments. Now, this is obviously quite limited because we can replicate what happens to a bunch of cells growing on a dish or a bunch of cells injected into a mouse, but a mouse is not a human. And the end goal of this is to create some meaningful clinical outcome or some indication that can be useful for treating patients. So the one step forward in adding one more layer of complexity, very similarly to what Fiona has presented, is, in my case, is the following, and that is to consider spatial interactions. So up to now, the model was basically saying that any of these 4 cell types can see any other cell type. So it doesn't matter where they're located physically in the space, any cancer cell is able to talk with certain frequency to any stroma cell, regardless of where it is. Obviously, this is not realistic in any way. So to make it more meaningful, we're going to introduce space explicitly as a component. And the way we do this is with something called hybrid discrete continuum model. The details of it doesn't matter, but basically it will be a 2D space on which I'm seeding a bunch of cells and I'm giving them individual rules of behavior. And their interaction will depend on where they are located in this 2-dimensional space. Now, this is basically an expansion of the diagram you saw earlier, but with just a bit more information. And this time, the cancer is not anymore either a sensitive or resistant cancer cell, but its behavior will be temporarily determined by a collection of environmental signaling that tells the cancer whether to go on and divide, maybe go in quiescent state, so become dormant, or die if conditions don't allow for growth. So all of these are pretty similar to earlier, but what we now have is environmental description of components that will determine the behavior of the cells. And these are governed by partial differential equations, very much similar to the ones you saw in the previous 2 presentations. So I'm not going to go through the talk to the equations. The main difference here is that now, as far as the individual cells go, they don't follow an actual equation like the ones you saw, but they basically follow some individualized kind of flow chart, decision chart. So each individual cell in the simulation will go through consecutive steps of decisions based on if there is space, if there's a condition to divide, and it will divide or would not divide and will die. And so this is sort of represented by these flow charts, and depending on the type of cell that I'm picking on, there will be a slightly different set of rules that the cell is following. And this all then comes into the bigger picture of this bigger model that will solve all of the dynamics at every point in time and space for individual cells, and also for those environmental conditions such as drug concentration. So visually, what this looks like is actually a 2D slice through a 3D tumor. And you can see there's some, well, the white will be empty, there's some cells here and there, and then the black will be some vessels. So you can imagine in the simulation that I'll show you earlier, these vessels are coming towards you out of the screen plane. And so they will be represented here by dots. And these will be the points to from where the drug comes out and is diffusing in tissue. So just to give you a quick understanding of why this is called hybrid discrete continuum model, the idea is basically that we have one description, which is the one with the cells. And so these will be different colors depending on the cell types, and they're spread all over this domain. But at the same time, on top of the very same domain, we're also solving these partial differential equations. And the coupling comes from the fact that the numerical grid on which we solve the partial differential equation corresponds to also to the grid on which we position the cells. And by doing so, the behavior of each individual cell will be determined by the amount, say, of drug at that very specific position. So I'm just going to show you a couple of movies just to conclude, but you'll see basically these three panels in a more interactive way. But what you're looking at is in the top left, the cell distribution. So in yellow are normal cells. So you can think of these as the normal tissue. So fibroblast, it will be either some empty space or some fibroblast cell in yellow. You see vessels spread all over. Even if you don't see them very well, you'll be able to notice where they are because you'll see the drug coming out of there. And then the cancer is either painted in red or in pink, or well, that looks more like orange if it is in a dormant state. So you know the red cells are actively proliferating and the other ones in pink or orange are dormant. So those are not proliferating. Now on the top right panel, you see the proliferation signal. So it's all normalized. So if there's 0, there's no proliferation signal. If it's maximum capacity 1, it means there's a lot in that position there of proliferation signal. And this is what effectively would determine the status or the fate, the decision of a cell to undergo cell death or stay in quiescent state or go on and proliferate. And then the bottom right panel finally will show you the drug. So this is when I mentioned that you'll actually see the position of the vessel because the drug is coming out of the vessel and then is being washed out from the very same location as sort of like a sink and source position. And again, the drug is governed by its own partial differential equation. Now, if we run this simulation, we can do many things. And one of them is to keep track of what happens to individual cells. So not only we have the bulk kind of summary statistic of all of these, for example, the tumor burden, which is immediately relatable to something that we observed in the clinic, like the curve that I showed you at the very beginning. But also, if we are curious to see why specific behaviors are occurring, we can go back in the simulation and check and figure out what is the kind of the history of that cell and how it got there and how it got to be resistant in that location specifically. So we can go a bit deeper about the reason of individual cell fate as it then emerges into the collective behavior of the bulk of the tumor that is growing or not growing. So I'll just show you a couple of simulation. And basically what I'm doing here is for this first simulation is for the entirety of it, going along the lines of like a few months. I'm just going to start the drug. So you'll see the drug coming in the bottom panel here. You'll see the drug is coming in, it will diffuse out, and then I'm keeping the drug on at all times. So you see initially there's the drug, there's an initial moment where it takes a while for the drug to actually diffuse across the whole domain. As soon as it does, you've seen a wave of sort of death coming into the tumor mass. And now you see these blue cells happening here that were not present at the beginning. So these will be normal fibroblasts that converted into reactive stroma upon the presence of sufficient amount of drug. And those are the drivers to the rebound. And in fact, you can see if you can follow the kind of the equivalent of the tumor burden curve here, corresponding to the presence of activated stroma, you see this rebound driven by the activated stroma. And eventually, the full tumor mass grows beyond the initial condition even. So this is quite unrealistic. In reality, we would interrupt the treatment way before getting here because clearly we make things worse. Now, with the kind of same mathematical setup, we can experiment what would happen now if we were to interrupt and pause the treatment. As I said, the good news is that if we pause the treatment for some drug holiday time, then the stroma will go back to renormalizing, and we'll have some kind of breathing time for the tissue, basically. So here, what I'm trying to, if you look at this kind of time plot here, you'll see the burst of drug being shown by these sort of blue windows. And here's just one example when I'm doing 2 weeks treatment on, 1 week treatment off. Carefully handpicked so that it actually looks good and I get a good result. But of course, behind this is lots of different attempts in more or less a systematic way. So what you see this time basically is you can first of all see from the targeted drug panel that the drug is effectively being pulsed. So it goes in and then it goes out. And diffusion does its job every time that the drug comes in or out. But the overall curve, even though it doesn't look as good at the beginning, so we're actually getting a lot of growth to begin with, which doesn't look ideal. Eventually, this pulsing on and off allows us to pretty much keep under control the whole tumor. And I say pretty much because we don't really get eradication. If you can see, there's these remaining pockets of resistance. And although this is sort of dynamical equilibrium, over time, we see that pretty much there's some locations that seem to favor some resistance overall. And then, of course, as I said, we can use these simulations to go back and investigate why were the conditions. So is it closest to the vessels? Is it presence of a given amount of help or in terms of stroma surrounding and all of that? So this is kind of a testing platform for all those questions, basically. And then, as I said, we can do a number of different things and we can try to vary parameters and actually ask the question, what is the best treatment schedule? This is by no way prescriptive because it's just a toy model, obviously. So this doesn't mean that they wouldn't go on and then apply the drug with this schedule, but it's just to show you how mathematics can put basically numbers into some equations that are formally and rigorously expressing what the dynamics are in reality and start trying to answer some of the questions. So I guess I don't have a concluding slide, I believe, but these will be kind of the overall picture, like putting it all together. And the idea is that now that we have the spatial interpretation, we can actually simulate multiple metastatic disease, which is often the case for patients that come in with metastatic disease, which is different locations of the tumor. And the idea is we can use that in simulation to find out what is the overall course of treatment for the patient to minimize growth across metastatic sites. So if there are any questions, otherwise I'll conclude.
Thank you so much, Noemi. That was, I think what you've seen across all the talks you've had is that diverse range that I was speaking about right at the beginning. Myself and Fiona started looking at sort of the mathematical intricacies, and Fiona mentioned about the idea of blow-up. There you really need your pure mathematics to say, okay, when can I solve these? When do they exist? What exists? How does it exist? But then you had all the way back to Noemi's work, where it was, we really have data that we want to understand and fit and predict. So absolutely fantastic. Thank you both for giving those talks. Like I say, I'll be putting this up later for those who couldn't make it, but if there are any questions from the room, please do put them in the chat or unmute yourself and we'd love to answer them.
Hi.
Hello.
So that was a very interesting talk. Thank you very much for that. So I was just wondering, what sort of modules would you say are absolutely essential for a career in mathematical biology? There
is no such thing as essential. If there is a mathematical biology course that you can take it, because that will give you a range of skills. But really, there's a new sort of field that's just emerging called topological data analysis in mathematical biology. Didn't exist before a few years ago, and that's from people coming from topology, coming in and saying, hey, we've got these skills, you've got data, maybe we can do something. And so, and that's where we're getting sort of the huge steps forward. It's people having skills that perhaps we don't have already, coming in and bringing them. So there are certain things you can do, your ODEs, PDs, that's what you saw today. Yeah. But really, don't restrict yourself. If you've taken a route where I've done number theory, I've got weird skills, there will be something you can do, I promise you. Okay. Fiona and Noemi, what do you think?
Yeah, I would agree 100% with what Thomas said. I think if there is mathematical biology courses, then they're definitely good, giving a good overview of different techniques in the field, but there's new areas coming up all the time, and using techniques from other areas to solve these problems is something that's really key.
Yeah, okay, thank you.
Absolutely. I think it's just more a matter of that if you take math bio courses, that they give you exposure to different things and maybe they give you ideas. But the technicality of things is whatever comes from an undergraduate degree, anything can be used.
Okay, brilliant. Thank you.
Not a problem.
And is there like any particular areas that you recommend for like extra reading that would be helpful?
I mean, there are two books called Mathematical Biology by Jim Murray, and they are the standard Bibles of mathematical biology. Like I say, mathematical biology as a field has grown since then, but they're two very nice, well-written books, which are sort of readable because they're Jim Murray. He wrote them as sort of anecdotal. Jim Murray was one of the very first mathematical biologists, and he could do whatever he liked. So you'll have one chapter on foxes spreading rabies across Europe, and then another chapter on maths of marriage, and then another chapter on crocodile teeth. He could do everything and anything he wanted. So it's a lovely book. Look, two sets of books. I firmly recommend them.
Okay, brilliant. Thank you.
Fiona, have you any other additions?
I was going to say the same books.
Yeah, no, they are very nice books.
It's a must. Yeah. And then I guess it depends. If you're specifically interested into something from there, then there are more specific ones. Obviously, the whole epidemiology thing has got their own.
Yeah. Oh, my epidemiology at the moment. There's a new book every week. Yeah. Yeah. So mathematical biology by Jim Murray, certainly a good start.
Okay, thank you.
Not a problem. And then if there are no more questions, I will kick you all out because I have to go lecture. Have a good day all. Thank you for coming along, and I hope that helped give you motivation and insight for your futures.